Hello students, welcome to Read MedPrep Academy channel. Today in Sexual Reproduction in Plants Part 2, we are going to discuss about an important pre-fertilization structure, the male reproductive part of a plant, the Androecium. Androecium is made up of stamens. Each stamen possesses an anther and a filament. The anther bears the pollen grains which represent the male gametophyte. The stamen which has a filament and an anther, the filament supports the anther, and the anther produces the pollen grains which develop the male gamete or the sperm. Now, what are the stages of development of anther? We are going to discuss one by one in our future lecture. One is Anther Primordium. Number 2 is Archisporial cell formation. Number 3 is division of the Archisporial cells. Number 4 is division of the primary parietal cells and the formation of the wall layers of the anther, formation of the sporogenous tissue. Number 6 is microspore mother cells and the formation of the microspore tetrads. And number 7 is the microspore formation. And number 8 is the pollen grain formation. Now, a very young anther develops as a homogeneous mass of the cells surrounded by an epidermis. This is called the Anther Primordium. During its development, the anther assumes a four-lobed structure. This is the first stage of the development. Here you can see a mass of cells surrounded by an epidermis. In each lobe, a row or a few rows of hypodermal cells below the epidermis becomes enlarged with the conspicuous nuclei. This functions as the Archisporium, and this is the stage of the Archisporial cell formation. Here you can see at the four corners, the Archisporial cell differentiation occurs. Now, the third stage is the division of the Archisporial cells. The Archisporial cells divide by periclinical divisions to form the primary parietal cells towards the epidermis and the primary sporogenous cells towards the inner side of the anther. Here you can see the Archisporial cell dividing on the outer side as the parietal cell and the inner side as the sporogenous cell. Now, the fourth stage is the division of the primary parietal cells. The primary parietal cells undergo series of periclinical and anticlinical division and form 2 to 5 layers of the anther walls, composed of the endothelium, the middle layers below the endothelium, and the tapitum below the middle layer from the periphery to the center. Here you can see the formation of the wall layers by the parietal cell into the endothelium, the middle layer, and the tapitum. The parietal cell layer divides below the epidermis and forms a single layer of the endothelium and 2 to 3 layers of the middle layer cells, and below the middle layer cells is the layer of the tapitum. What is microsporogenesis? The stages involved in the formation of the haploid microspores from the diploid microspore mother cell through the meiotic divisions is called microsporogenesis. Now, the stage 5 is the formation of the sporogenous tissue, the start of the microsporogenesis. The primary sporogenous tissue cells directly or may undergo a few mitotic divisions to form the sporogenous tissue. This is the sporogenous stage. Here you can see below the tapitum, the sporogenous cells multiplies and forms a mass of the sporogenous tissue. The stage 6 is the microspore mother cell formation and the formation of microspore tetrad formation. The large generation of the sporogenous tissue functions as the microspore mother cells. Each microspore mother cells divides meiotically to form a tetrad of 4 haploid microspores. This is called the microspore tetrad or the pollen tetrad stage. Here you can see inside below the tapitum formation of the pollen tetrads or the microspore tetrads. The stage 7 is the microspore formation. The microspores soon separate from the tetrads from one another and remain free in the anther locule. Here you can see in this image the microspore tetrads separate and form the individual microspores. This is the microspore stage. Now, these microspores mature themselves in the locule and develop into the mature pollen grains. Here you can see the formation of the pollen grains within the anther cavity. Now, to sum up the stages of development of Microsporangia, the first stage is the development of the Anther Primordium. The second stage is the differentiation of the Archisporial cell. Third stage is the formation of the Peritoneal sporogenous cell from the Archisporial cell. And the fourth stage is the formation of the different wall layers by the parietal cell. The fifth stage is the division of the sporogenous cell into multiple sporogenous cells and the sporogenous mother cells. And the sixth stage is the formation of the pollen tetrads from the microspore mother cells and the formation of the microspores by the separation of the pollen tetrads. And the last is the maturation of the microspores into the pollen grains. Now, what is Pollinium? In some plants, all microspores in a sporangium remain held together, called Pollinium. Example, Calotropis. Now, what is the structure of the Pollinium? Pollinia are attached to a clamp or a clip-like structure, sticky structure called the corpusculum. And the filamentous or thread-like part arising from each Pollinium is called the retinaculum. The whole structure looks like an inverted Y-shaped structure and is called a translator. Here you can see the Y-shaped structure, the Pollinium attached to the retinaculum by the cortical and the retinaculum attached to the sticky structure of the corpusculum. Now let us discuss the transverse section of the mature anther. The transverse section of the mature anther shows an anther wall. Inside the anther wall is the anther cavity. Anther is bilobed. Each lobe has two theca, hence it is called dithecous. A typical anther is tetrasporangiate. That means it has 4 sporangia. The transverse section will show the anther wall, the anther cavity and the connective tissue connecting the sporangia. In this structure, you can see the connective tissue in the center having the vascular bundle which is continuous with the filament, and the anther wall which is made up of the epidermis outside. Inside to the epidermis is the endothecium. Inside the endothecium is the middle layers which are 2 to 3 in number, and inside the middle layer is the tapetum, and in between the sporangia the endothecium is very thin and may dehisce, and this area is called the stomium. This helps in the release of the pollen grains from the sporangia. Now let us discuss the anther wall. The mature anther wall consists of the epidermis, the endothecium, the middle layers and the tapetum. Here you can see in this image the different layers from the epidermis. Inside is the endothecium. Inside are 2 to 3 layers of the middle layers, and inside to the middle layers is the tapetum. Inside the tapetum the sporogenous tissue consisting of the microspore mother cells. Now epidermis, it is a single layer and protective in function. The cells undergo repeated anticlinical divisions to cope up rapidly with the enlarging size of the tissues. Now endothecium, it is generally a single layer of radially elongated cells. You can see in the image how the cells are radially elongated below the epidermis. The inner tangential wall develops bands, sometimes radial walls also, of alpha cellulose. Sometimes they are also lignified. The endothecium is hygroscopic, and in the anthers of aquatic plants, saprophytes, clistogamous flowers and extreme parasites, endothelial differentiation sometimes may be absent. Now the cells along the junction of the 2 sporangia of an anther lobe lack the thickenings, and this region is called the stomium. Why the stomium is important? Because of the hygroscopic nature of the endothecium, it helps in the dehiscence of the anther at the site of the stomium after the maturity of the pollen grains. Here you can see in this image the stomium, and inside the stomium are the large number of pollen grains, and the pollen grains are released through the dehiscence of the stomium. Now the middle layers of the anther wall. 2 to 3 layers of cells next to the endothecium constitute the middle layers. They are generally ephemeral. They disintegrate or get crushed during maturity. The tapetum, it is the innermost layer of the anther wall and attains its maximum development at the tetrad stage. Now, it is considered that the tapetum is dual in origin. Partly it is derived from the parietal cells and partly from the connective tissues. And thus the tapetum is considered to be dual in origin. You can see from the image and understand it. The nucleus of the tapetum is usually single or maybe more than one nucleus, or the nucleus may become polyploid. The tapetum nourishes the developing sporogenous tissue inside mother cells for their development. Tapetum also contributes to the production of the wall materials like the sporopollenin, pollen kit, tryphin, and number of proteins that control the incompatibility reaction. Tapetum also controls the fertility and or sterility of the microspores or pollen grains. There are two types of tapetum based on its behavior. They are secretory tapetum and invasive tapetum. Secretory tapetum is also called parietal tapetum or glandular tapetum or cellular tapetum. Tapetum retains the original position and cellular integrity and nourishes the developing microspores. In this image, you can see the early stage of the tapetum forming the secretory tapetum, which helps in the production of various proteins and nourishes the maturing of the pollen grains. Invasive tapetum is also called periplasmodial tapetum, where the cells lose their inner tangential and radial walls and the protoplast of the tapetal cells coalesce together to form a plasmodium. Many botanists speak of a third type of tapetum called the amoeboid tapetum, where the cell wall is not lost. The cells protrude into the anther cavity and move into the anther cavity through an amoeboid movement. This type is often associated with the male sterility and should not be confused with the periplasmodial type. Here you can see on the left side the early stage of the tapetal cells, and these cells separate themselves from the tapetal wall and move into the anther cavity by amoeboid movement and form the amoeboid tapetum. Now, what are the functions of tapetum? It supplies nutrition to the developing microspores. It contributes sporopollenin through ICSCE bodies, thus plays an important role in the pollen wall formation. Here you can see that the sporopollenin is formed by the lipid metabolism, and this sporopollenin forms the ICSCE bodies inside in the anther cavity. What are ICSCE bodies? ICSCE bodies are the small acellular structure of the sporopollenin. They are present in cells of the tapetum and they help in the development of the pollen grains. They are of lipid nature. They appear in the cytoplasm of the tapetal cells during the spore wall formation. Then they migrate to the cell surface where they are released into the anther cavity. ICSCE bodies are also involved in the external thickening of the exine because of the presence of sporopollenin. Now the third function of tapetum is the pollen kit material, which is contributed by the tapetal cells and is later transferred to the pollen surface. Number 4 is the exine proteins, which are responsible for the rejection reaction of the stigma, are present in the cavities of the exine. These proteins are also derived from the tapetal cells. Now let us discuss about the anther cavity. The anther cavity is filled with microspores at the young stages. And as the sporangia develop, the anther cavity is filled with the pollen grains. The meiotic division of the microspore mother cells give rise to the microspores, which are haploid in nature, and the maturation of the microspores form the pollen grains. Now something about the connective tissue. It is a column of the sterile tissue surrounded by the anther lobe. It possesses vascular tissue which is continuous with the filament. It also contributes to the inner tapetum. Here in this image, you can see that the tissues in between the sporangia is the connective tissue, and in the center of the connective tissue, you can see a vascular bundle which connects with the filament. Microspores and pollen grains. Microspores are the immediate product of meiosis of the microspore mother cell, whereas the pollen grain is derived by the maturation from the microspores. The microspore have protoplast surrounded by a wall which is yet to be fully developed, whereas the pollen protoplast consists of the dense cytoplasm with a centrally located nucleus. Now, let us talk about the wall of pollen. The wall is differentiated into two layers. The inner layer is called the intine, which is thin, and the outer layer is thick and called exine. Intine is thin, uniform, and is made up of pectin, hemicellulose, cellulose, and callose together with proteins. Exine is thick and is made up of cellulose, sporopollenin, and pollen kit. The exine is not uniform and is thin at certain areas. When these thin areas are small and rounded, it is called germ pores, or when elongated, it is called furrows. Exine germ pores, it is associated with germination of pollen grains. The sporopollenin is generally absent in the germ pores. You can see in the image the exine lacking the sporopollenin at the areas of the germ pore. Now, what is the composition of the substances that are present in the intine and exine? In the exine, the pollen kit is present, which is made up of a fatty substance, and sporopollenin is present. Whereas the intine is made up of cellulose, hemicellulose, and callose. The surface of exine is either smooth or sculptured in various patterns. Rod-like, grooved, warty, or punctuate, etc. The sculpturing pattern is used in the plant identification and the classification. Here you can see the different surfaces of the pollen grain formed by the exine. On the left, number 1 is the knobby structure, number 2 is the needle structure, number 3 is the net structure, number 4 is the fingerprint structure, number 5 is the combination of the needle and the net structure, number 6 is the combination of the knobby and the needle structure. Now, what is the shape of the pollen grain? The shape of the pollen grain varies from species to species. Few of them may be globose, ellipsoid, fusiform, lobed, angular, crescent shape, etc. In this image, you can see the different shapes of the pollen grains. Globose, ellipsoid, elongate, pyriform, obpyriform, ovoid, obovoid, lemoniform, turbinate, obturbinate. So how the shapes look like of the pollen grains you can see. What is the size of the pollen? The size of the pollen varies from 10 micrometers in Myosotis to 200 micrometers in members of the family Cucurbitaceae and Nyctaginaceae. Now, something about the pollen kit. Pollen kit, you know that it is derived from the lipids and is contributed by the tapetum and is colored yellow or orange and is chiefly made up of carotenoids or flavonoids. It's an oily layer forming a thick viscous coating over the pollen surface. It attracts insects and protects from the UV radiation. Now, let us discuss of the development of the male gametophyte. The microspore is the first cell of the male gametophyte and is haploid in nature. The development of the male gametophyte takes place while they are still in the sporangium. Here you can see the first development that occurs is the vacuolation of the pollen grain. Then the division of the nucleus occurs in the microspore meiotically to form the vegetative nucleus and a generative nucleus. Here you can see in this image the division of the nucleus. And after the division of the nucleus, a wall is laid around the generative nucleus, resulting in the formation of two unequal cells. A large irregular nucleus bearing with abundant food reserve called the vegetative cell and a small generative cell. Here you can see the vegetative cell and the generative cell in the microspore. Generally, at this two-cell stage, the pollens are liberated from the anther. In some plants, the generative cell again undergoes a division to form two male gametes. Here you can see the bicellular pollen. It has a generative cell and a vegetative cell. In a tricellular pollen, the generative cell undergoes division and forms two generative cells, and there is one vegetative cell. It is a tricellular pollen. The pollen is liberated at the two-cell stage. In 60% of the angiosperms, pollen is liberated in two-cell stage. Here you can see the splitting of the stomium and the dehiscence of the anther and the release of the pollen grains. Further, the growth of the male gametophyte occurs only if the pollen reaches the right stigma. The pollen on reaching the stigma absorbs moisture and swells. The intent grows as pollen tube through the germ pore. Here you can see the pollen stigma interaction. The pollen falls on the stigma and the pollen is captured by the stigma. The pollen and the stigma adhesion occurs and there is absorption of moisture by the pollen grain, and this causes the germination of the intine and forms a pollen tube which penetrates the stigma and enters into the style, and this pollen tube reaches the ovary. Here you can see during the development of the pollen tube will have a vegetative nucleus called the tube nucleus and a generative nucleus. In the pollen tube, the generative nucleus starts dividing to form two male nuclei. In the case of the pollen is liberated at a two-cell stage, the generative cell divides in the pollen into two male cells or the sperm cells after reaching the stigma or in the pollen tube before reaching the embryo sac. Here is the summary of the stages in the development of the male gametophyte. The first stage is the vacuolation of the pollen grain and the division of the nucleus into the vegetative nucleus and the generative nucleus, formation of the vegetative cell and the generative cell, and the pollen grain reaches the stigma and there is absorption of moisture and development of the pollen tube. In the pollen tube, there is division of the generative nucleus into two male gametes. Now, let us move on to palinology. What is palinology? Palinology is study of pollen grains. It helps to identify the distribution of coal and to locate oil fields. Pollen grains also reflect the vegetation of an area. Palinology is used in phylogenetic analysis. It can be utilized in plant identification. Extant plants, fossil plants by paleopalinology or paleobotany, past plant communities, the climatic variations that had occurred, biogeography and migration of plants, all these have been studied by palinology. Liquid nitrogen minus 196 degrees centigrade is used to preserve the pollen in viable condition for prolonged duration. This technique is called cryopreservation. Cryopreservation is used to store pollen grains in pollen banks of economically important crops for future breeding programs. Pollen banks are used to store the pollen grains for a short as well as a very long period of time in a viable condition. The very important application of pollen bank is to preserve the biodiversity in the form of preservation of valuable of genetic resources. Now, let us talk about the bee pollen. Bee pollen is a natural substance and contains high protein, carbohydrates, trace amount of minerals and vitamins. Therefore, it is used as a dietary supplement and is sold as pollen tablets and syrups. Bee pollen increases the performance of athletes, race horses, and also heals the wounds caused by burns. The study of honey pollen is called melitopalinology. Now, what are the benefits of bee pollen? It increases the longevity by neutralizing the free radicals. It is an effective aphrodisiac, increases muscle, helps clear acne. It has a high nutritive value, may help to improve fertility, increases strength, endurance and energy, reduces the production of histamine which makes it a good remedy for allergies, can help to reduce or reverse aging and wrinkling, assist in reducing prostate problems. So today in Sexual Reproduction in Plants Part 2, we discussed an important pre-fertilization structure, the Androecium, which is the male reproductive part of a plant. We also talked about palinology and bee pollen. Thank you. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Kindly log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.